All right, so our last presenter in this section will be ZATA Pharmaceuticals.
I'm David Tabatadze, president of ZATA Pharmaceuticals, and it's my pleasure to be here and to tell you about ZATA's donated blood purification closed systems. I think, previous presenters scared you quite a bit, but I will try now to show products that saves a lot of lives from drug-resistant pathogens. So there is a danger when you have blood transfusion. You can be contaminated with Ebola, HIV, Zika, but to prevent that happens, we are currently using a large number of tests. Now, a high number of tests means high cost for blood centers. That's unsustainable for them, and so let me show you dynamics, how number of tests went up. If 90s, it was 4 tests for $30. Last year, it was 18 tests, close to $150. And for 2025, is predicted to be 20 to 24. That will be close to $150. And it is not only unsustainability of cost, but it's also not 100% safe. Now that problem has been acknowledged by FDA and NIH, and currently they are looking technology that will inactivate all pathogens such as virus and bacteria in donated blood, that bring cost down and also makes product safer. And actually that is what our technology is all about. And let me show what our product looks like. Here you are collecting blood by needle from the donor. In the black, number 2, we are adding active compounds that eliminates all presented pathogens. After the treated blood is purified through cartridge, and sterile blood is ready for use. Obviously, we are not only one player in the field. There is 2 other companies. One is Terumo in Tokyo, Japan. Another one, Cirrus in Concord. But difference is that their products only works in clear substances such as plasma and platelets, versus ours that works in all blood components. On top of that, it's very cost effective, and as you've seen in previous pictures, could be used in non-clean room environments, including in the field. So now, after familiarity with our technology and science behind it, a couple of well-established institutions in the field has considering partnership. One of them is already became our partners 1 month ago. It's New York Blood Center, one of the largest blood centers in the nation. Second one is multinational European pharmaceutical company, and we haven't finalized the partnership deal yet, and therefore cannot disclose. So our product, we anticipate to get on market in 2025, and our first customers will be identified partners. They each can sell up to 1 million units according to their calculation, and anticipated revenue will be around 300 million. Total market in United States and the European Union is around 4 billion. And considering cost effectivity, we again provide profit could be close to 3 billion. So we are done. Discovery stage currently in preclinical, patent protecting until 2035, and currently looking around the investment. Here is the brain power behind our product. Now, for those who had hard time to understand my Georgian accent, on this slide I summarized what I just presented. So I want to thank to MBI, NIH for support, and ZATA's team for their great contribution, and we'll be more than happy to answer your questions.
So first, I just want to recap in my poor, meager English to make sure I understood what you said. So you have, to grossly oversimplify, made a filter. So put any blood through, and it's going to filter out the pathogen. So let me just stop. Did I hear you basically correctly?
Not entirely. Procedure is we are adding in a blood small compounds that inactivates pathogens. Then we are running treated blood through cartridge extracts whatever we added in it. So the residual compounds and
But dirty blood goes in one side, clean blood comes out the other side.
no, blood entirely go through, and we are only catching what we added. And if there was pathogen is inactivating. So you are getting pure blood,
Okay.
and you do not need to use tests after that. So test will be eliminated when technology is approved.
And I know it's very early, but you have some initial indicators of, so people are going to be spending $250 on your main competition, which is testing the heck out of it and hoping you haven't missed something. Your thing is going to cost $200, $20, $2.
Way less than that. So actually, each test costs roughly $10. So if we sell our unit, let's say 150, that will be 5 times higher than manufacturing costs. So considering you do not need tests, you do not need development future tests.
Okay, so this thing's really cheap to manufacture is what I think I just heard you
Yes,
say.
that's very, very cheap, yes.
And it's still going to be a lot cheaper than the alternative. Okay, sorry, I'm hogging the microphone next.
Could you talk a little bit about the results that you've achieved, and if they're with live animals or any, how far have you gotten in the testing?
Yes. So what we've done so far, we added blood, all kind of pathogens, bacteria, viruses, gram-negative, gram-parasites, and we inactivate them entirely. So nothing survives. Another de-risking test, what we've done was again, let's make one statement that our compound is not pathogen specific, it's a genomic molecule specific. Every germs you name, they have a DNA of RNA. So our compounds gets inside and destroys these bugs that way. So what we done for this, the de-risking, we done reactions with a lot of surface proteins. Our compounds do not modify anything but DNA, RNA, and so animal studies has not been done, but a lot of de-risking on models in terms of inactivation, pathogens, and we also do some model molecules. That's so far the risking we've done.
I had a question on your leadership team. So it's clear you got crazy awesome scientists on board. It's clear you've got partners that are the kind of partners I like to see. But of course, to sell the product, you need people that have deep experience, because the healthcare system, wicked easy to sell into, right? Totally a breeze. So can you tell us a little bit about the people you've lined up or are lining up to help you build the financial infrastructure to be able to actually market and sell this thing when it's ready?
So about financial chairman of board of directors happens to be former chief financial officer, publicly traded company, and he is even currently also chief financial officer for publicly traded company. That said, we still are in search business person or persons who could help us business development model. And if somebody presented here wants to be part of our success, or you have some good candidate, more than happy to talk.
I jumped in front of your question, sorry.
So, and regarding of experts, we definitely have experts, scientific experts, we have no problem, we have more than we need.
I just wanted to clarify where you were in the along the way. So you had a slide up. You went by it pretty fast. But so where exactly are you in development of the product?
So currently we are advanced preclinical development. So we just started collaboration in preclinical development with New York blood centers. It's not only large in terms of the supplying of blood, they also have a strong scientific team. And we also received maybe largest grant in the field, not only in Worcester, maybe in Massachusetts, $3 million. So it will take us to finish preclinical development in 2 years. Hopefully, if we are also looking for round A investments, and it could be much faster done if we will get more resources.
Well done. Thank you very much.